Welcome to Nexus Clinical. Before diving into this lecture, I want to ensure we're all on the same page. This is for general informational purposes only and does not constitute the practice of medicine, nursing, or other professional healthcare services, including the giving of medical advice. No doctor-patient or pharmacist-patient relationship is formed. Using this information and the materials linked to this content is at the user's risk. This is not intended to substitute for professional medical advice, diagnosis, or treatment. Users should not disregard or delay seeking medical advice for any medical condition they have, and they should consult their healthcare professionals for any such conditions. Clinical experts created the references, content, and clinical insight. NotebookLM, a Google AI tool, created the content, which I extensively reviewed before release. Finally, the host states that he takes all conflicts of interest seriously. Currently, there are no conflicts to disclose. For all of his disclosures and the companies he invests in or advises, he directs users to reach out independently, where he keeps an up-to-date and active list of all disclosures. The field of genetic medicine is advancing rapidly from theoretical promise to clinical practice through strategies like gene replacement, which adds functional genes, and gene editing, which makes precise DNA changes. In vivo treatments primarily rely on adeno-associated virus, AAV, vectors, enabling approved therapies like Luxturna, Zolgensma, Elevidys, and Beqvez. These successes required overcoming AAV limitations like small capacity and immune responses. Next is in vivo gene editing, using tools like CRISPR/Cas to permanently fix DNA, surpassing older methods like ZFNs and TALENs. This technique exploits DNA repair pathways, including NHEJ for knockouts and HDR for precise edits. Delivery methods are evolving. Liver-focused therapies now favor lipid nanoparticles, LNPs, over AAVs, reducing long-term off-target risks. Advancing this field demands addressing challenges such as off-target effects, immune responses, scalable manufacturing, and ethical issues related to permanent genome editing. The following learning objectives are designed to capture the core concepts, mechanisms, and clinical implications of in vivo genetic medicine. First, students should be able to differentiate between the therapeutic goals of gene replacement, which involves gene addition, and gene editing, which encompasses genome correction or disruption. They should also be able to distinguish between in vivo and ex vivo delivery modalities. Next, learners should understand the structure and mechanism of transduction for the adeno-associated virus vector. This includes how the genome persists as a stable apisome in non-dividing cells, enabling durable, long-term expression of therapeutic genes. It is important to identify the two major limitations of the adeno-associated virus platform: its limited packaging capacity of approximately 4.7 kilobases and the presence of pre-existing immunogenicity. These constraints have driven the development of novel solutions, such as the microdystrophin gene used in the therapy Elevidys. Furthermore, students should analyze how the strategic selection of adeno-associated virus serotypes is employed to achieve tissue-specific targeting. For example, AV2 is used to target retinal pigment epithelial cells in the treatment Luxturna, while AV9 targets central nervous system motor neurons in Zolgensma. Learners should also be able to outline the critical risk mitigation strategies necessary for systemic gene therapies, including the use of prophylactic systemic corticosteroids to manage immune-mediated liver toxicity and the importance of pretreatment screening for neutralizing antibodies. In addition, students should compare and contrast the three major nuclease platforms used in gene editing technologies. These include zinc finger nucleases, transcription activator-like effector nucleases, and the CRISPR/Cas9 system. The role of the RNA-guided mechanism in CRISPR/Cas9 makes it the preferred and highly programmable tool in gene editing applications. Students should differentiate between the error-prone non-homologous end joining and the highly precise homology-directed repair pathways. They need to understand how nucleases exploit these cellular pathways to achieve gene knockout or precise correction or insertion, respectively. Further, learners should justify the strategic shift toward using lipid nanoparticles instead of adeno-associated virus vectors for many in vivo gene editing therapies targeting the liver. This shift is based on the nanoparticle's ability to provide the transient expression necessary to minimize off-target effects. Lastly, students should identify off-target effects, which involve cuts at unintended genomic locations, and on-target effects, such as unintended large deletions. These are primary safety concerns in gene editing, emphasizing the need for rigorous efforts to engineer high-fidelity Cas enzymes. They should also differentiate between somatic gene editing, which involves modifications in non-reproductive cells, and germline gene editing. It is important to discuss the ethical imperatives for ensuring safety, informed consent, and equitable access to these technologies, which have the potential to offer permanent cures. The field of genetic medicine has entered a transformative era, shifting from theoretical promise to clinical practice. At its core, it aims to treat or cure diseases by modifying the genetic material inside a patient's cells. This is achieved through two main strategies: introducing a functional gene copy to replace a defective one, gene replacement, or making precise, permanent edits to the existing DNA sequence, gene editing. The delivery of these genetic materials can be done through two primary methods, in vivo and ex vivo, a choice mainly driven by the biological nature of the disease and the accessibility of the affected tissues. The first wave of approved therapies, especially those delivered in vivo, has been predominantly made possible by a single, versatile platform, the adeno-associated virus, AAV vector. This introduction will define these fundamental concepts, laying the groundwork for understanding the approved therapies and the future of genomic medicine. The primary distinction in the administration of gene therapies lies in where the cellular modification occurs, inside or outside the patient's body. In vivo, within the living, gene therapy involves the direct administration of the therapeutic genetic material into the patient. This is typically achieved through a systemic intravenous infusion or a localized injection directly into the target organ or tissue. The genetic modification of the target cells thus occurs entirely within the patient's body. This approach is indispensable for treating diseases affecting solid or internal organs that cannot be easily removed and returned, such as the liver, central nervous system, CNS, muscle tissue, and the eye. Ex vivo, outside the living, gene therapy, also known as gene-modified cell therapy, is a multi-step process. First, target cells, most commonly hematopoietic stem cells, HSCs, from the blood or bone marrow, are harvested from the patient. These cells are then sent to a specialized laboratory where they are genetically engineered using a vector to introduce the therapeutic gene. After modification, and often after the patient undergoes a conditioning regimen like chemotherapy to clear existing stem cells, the engineered cells are infused back into the patient's body. This approach is predominantly used for hematological and immunological disorders where the target cells are readily accessible and can be effectively manipulated outside the body. An advantage of the ex vivo method is its ability to verify genetic modifications and assess cell health before returning them to the patient, offering a higher degree of control and potentially reducing the risk of off-target effects within the body. However, it is a complex, time-consuming, and expensive process that is less scalable than in vivo approaches. Beyond the delivery modality, the therapeutic strategy itself can be broadly categorized into two main types: gene replacement and gene editing. Gene replacement, often called gene addition, is the main approach of modern gene therapy. This method targets monogenic diseases caused by loss-of-function mutations by adding a new, functional copy of the affected gene into the target cells. The therapy does not change the patient's existing mutated gene. Instead, it provides an extra genetic template that enables the cell to produce the missing or faulty protein, restoring normal cell function. This form of gene therapy, GT, has now achieved notable clinical and regulatory success, with many approved products showing its therapeutic effectiveness. All currently approved in vivo AAV-based therapies work on this principle. Gene editing, also known as genome editing, is a more advanced and precise therapeutic strategy. Unlike simply adding a new gene, gene editing aims to directly modify the existing DNA sequence within a cell's genome. This is achieved using molecular tools programmed to locate specific regions in the DNA and make exact changes. The potential applications of gene editing are broad. They include repairing mutations that cause diseases, which involves restoring a disease-causing mutation to its normal sequence. It can also involve disabling a gene that produces a toxic or unwanted protein, an action known as gene knockout or disruption. Additionally, gene editing can insert new DNA sequences, such as therapeutic genes, at specific targeted locations within the genome, a process known as gene knock-in or insertion. While gene editing offers great potential for a permanent, curative treatment, it is still in earlier stages of clinical development compared to gene replacement, especially for in vivo applications where the editing tools must be delivered directly to cells inside the body. The clinical success of in vivo gene replacement therapy has been almost entirely dependent on the development and refinement of the adeno-associated virus, AAV, as a gene delivery vehicle or vector. AAVs have emerged as the leading platform due to a combination of favorable biological properties. Adeno-associated viruses are small, non-enveloped particles with a diameter of approximately 25 nanometers. They contain a single-stranded DNA genome that is about 4.7 kilobases in length. Wild-type adenovirus-associated viruses are not known to cause disease in humans and generally provoke only a mild immune response. For therapeutic applications, recombinant versions of these viruses are engineered to be replication deficient, which enhances their safety profile. This is achieved by removing the genes responsible for the virus's ability to replicate and form its protein shell, known as capsid. These removed genes are then replaced with a therapeutic genetic construct, a cassette, that includes the gene intended for treatment, a promoter to control the gene's expression in target cells, and a polyadenylation signal to terminate transcription. The entire construct is flanked by special sequences known as inverted terminal repeats. These repeats are the only viral elements retained because they are crucial for packaging the genetic material into the virus's protein shell and for processing the genetic material within the host cell nucleus. The process of AAV-mediated gene delivery begins when the viral capsid binds to specific receptors on the surface of a target cell, initiating entry via endocytosis. The AAV particle is then transported through the cytoplasm to the nucleus, where it uncoats and releases its single-stranded DNA genome. A critical and often rate-limiting step for transgene expression is the conversion of this single-stranded DNA into a stable, double-stranded form. Once this occurs, the therapeutic gene can be transcribed and translated by the host cell's machinery. In non-dividing cells, such as neurons, muscle cells, and retinal cells, the RAAV genome primarily persists as a non-integrating, circular DNA molecule known as an apisome. This stable apisome serves as a durable template for long-term protein expression, often lasting for years after a single administration. A key advantage of the adeno-associated virus platform is the natural diversity of its capsid proteins. There are over 100 different variants and serotypes of these viruses, each with a unique capsid structure that influences its affinity or tropism for different cell and tissue types. This diversity enables the selection of an optimal serotype to target a specific organ affected by a disease. For example, the serotype known as adeno-associated virus type 2 is particularly efficient at transducing retinal pigment epithelial cells. Because of this, it is often used in ocular gene therapies, such as Luxturna. Another serotype, adeno-associated virus type 9, has a remarkable ability to cross the blood-brain barrier, making it suitable for treating neurological disorders like spinal muscular atrophy, as demonstrated by the therapy Zolgensma. Additionally, adeno-associated virus Rh74 exhibits strong tropism for skeletal and cardiac muscle tissue, and it is utilized in the treatment of levities for Duchenne muscular dystrophy. AAVs are regarded as some of the safest and most effective platforms for gene delivery in vivo because they are non-pathogenic, can transduce non-dividing cells, and can produce long-lasting transgene expression from a single dose. However, the platform faces two major limitations that have significantly impacted the design and application of AAV-based therapies. The AAV capsid can only carry a genome of approximately 4.7 kb. This limited capacity acts as a major restriction, preventing the delivery of large genes. For example, The gene for dystrophin is too large to fit into a standard AAV vector, which has led to innovative bioengineering approaches for treating Duchenne muscular dystrophy. As AAVs are common viruses, a substantial portion of the human population, ranging from 40% to over 70%, depending on the serotype, has pre-existing neutralizing antibodies, NAbs, resulting from prior natural exposure. These NAbs can bind to the AAV capsid upon administration, hindering its ability to reach target cells and making the therapy ineffective. This presents a major reason for patient ineligibility for AAV-based treatments and remains a significant challenge in the field. These fundamental constraints have created a dynamic tension in the field, where the most effective available tool for in vivo gene delivery possesses inherent flaws that must be overcome. This challenge has become a primary driver of innovation. The need to circumvent the packaging limit led directly to the rational design of the microdystrophin transgene used in Elevidys, a shortened but functional version of the dystrophin gene that can fit inside the AAV vector. Similarly, the imperative to manage the host immune response has spurred the development of sophisticated clinical protocols. These include the mandatory use of prophylactic systemic corticosteroids with therapies like Zolgensma and Elevidys to suppress immune reactions against the vector capsid and the transgene product. It has also made pre-treatment screening for NAbs a critical eligibility requirement for therapies such as Begvez and Elevidys, ensuring that the treatment is only given to patients who are likely to respond. Therefore, understanding the interplay between the advantages and limitations of the AAV platform is essential for appreciating the design choices, clinical challenges, and future research directions for all AAV-based therapies. The successful translation of AAV vector technology from the laboratory to the clinic has resulted in the regulatory approval of several landmark therapies. These products represent the first generation of in vivo genetic medicines, each tailored to address a specific monogenic disease by replacing a single faulty gene. An in-depth analysis of these therapies, Luxturna, Zolgensma, Elevidys, and Begvez, reveals the versatility of the AAV platform and illuminates the scientific and clinical strategies employed to overcome distinct biological challenges. Luxturna, Veretagene Nepervovec RZYL, approved by the FDA in 2017, was the first gene therapy approved in the U.S. for an inherited disease, marking a historic milestone for the field. Luxturna is approved for treating patients with confirmed biallelic RPE65 mutation-associated retinal dystrophy. This group of autosomal recessive disorders, which includes subtypes of Leber congenital amaurosis, LCA, and retinitis pigmentosa, RP, is marked by severe, progressive vision loss that usually results in total blindness in young adulthood. The defect stems from the RPE65 gene, which encodes an isomerohydrolase enzyme vital for the visual retinoid cycle. Located in the retinal pigment epithelial RPE cells, this enzyme converts all transretinal ester into 11-cis-retinol. The 11-cis-retinol then binds to opsin proteins in photoreceptor cells to form visual pigments. Mutations in RPE65 lead to decreased or absent enzyme activity, blocking the visual cycle and preventing the regeneration of these pigments, which ultimately causes photoreceptor cell death and vision loss. An essential condition for treatment is the presence of viable retinal cells, as the therapy cannot restore cells that are already lost. Luxturna is a gene augmentation therapy that provides a functional copy of the human RPE65 gene to RPE cells. It uses an AAV2 vector known for its high efficiency in transducing RPE cells and its low likelihood of causing inflammation in the eye's immune-privileged environment. The therapy is given through a single surgical subretinal injection, which delivers the vector directly to the targeted RPE cell layer. Once transduced, these RPE cells use the new genetic instructions to produce functional RPE65 protein, restoring the visual cycle and enhancing the patient's ability to detect light. The approval of Luxturna was based on a comprehensive clinical program, including a pivotal Phase 3 randomized, controlled trial. The primary endpoint was the change from baseline in the bilateral multiluminance mobility test, MLMT, score at 1 year. The MLMT is a validated, performance-based test that assesses functional vision by requiring patients to navigate a standardized course with obstacles under 7 different levels of illumination, ranging from office-level lighting, 400 lux, to very low light, 1 lux, simulating real-world navigation challenges. At 1 year, patients treated with Luxturna demonstrated a statistically significant and clinically meaningful improvement in their ability to navigate at lower lux levels compared to the control group. The median MLMT score change was 2 in the treatment group versus 0 in the control group. Furthermore, 65% of participants in the intervention group successfully completed the MLMT at the lowest light level of 1 lux, whereas no participants in the control group could do so. Significant improvements were also observed in a key secondary endpoint, full field light sensitivity threshold, FST, testing, with the treatment group showing more than a 100-fold increase in light sensitivity. Follow-up studies have shown that these improvements in functional vision last, with benefits maintained for at least 8 to 9 years after treatment. Luxturna is administered surgically by an experienced intraocular surgeon. The treatment involves a vitrectomy followed by the subretinal injection of 0.3 milliliters of diluted vector suspension. Each eye is treated on a separate day, with at least 6 days between procedures. To manage potential inflammation, patients take a 7-day course of systemic oral corticosteroids starting 3 days before the first injection. The most common adverse reactions are related to ocular surgery and include conjunctival hyperemia, cataract development or progression, increased intraocular pressure, and retinal abnormalities such as retinal tears, macular holes, and subretinal deposits. Approved by the Food and Drug Administration in 2019, Zolgensma revolutionized the treatment landscape for spinal muscular atrophy, which is one of the leading genetic causes of infant mortality. Spinal muscular atrophy is a severe and progressive neuromuscular disease caused by mutations in both copies of the survival motor neuron 1 gene. The absence of a functional survival motor neuron 1 gene leads to a systemic deficiency of the survival motor neuron protein, which is essential for the survival and proper function of motor neurons located in the spinal cord. Without sufficient survival motor neuron protein, these motor neurons degenerate and die, resulting in irreversible loss of motor function, progressive muscle weakness and atrophy, and paralysis. In the most severe form of the disease, which can appear in early infancy, if left untreated, it can lead to respiratory failure and death, usually by the age of 2. Humans also have a nearly identical backup gene known as survival motor neuron 2. However, due to a single nucleotide difference that affects the gene splicing process, this gene primarily produces an unstable, truncated protein and only about 10% of the full-length, functional survival motor neuron protein. Zolgensma is a gene replacement therapy designed to directly target the monogenic cause of SMA. It uses an AAV9 vector to deliver a fully functional copy of the human SMN gene into the patient's cells. The AAV9 serotype was specifically selected for its unique ability to cross the blood-brain barrier and transduce motor neurons across the CNS after a single systemic intravenous infusion. Once the vector reaches the motor neurons, the delivered SMN gene functions as a stable apisome, allowing the cells to continuously produce the vital SMN protein. This aims to preserve existing motor neuron function, halt the disease's progression, and support the development of motor milestones. The efficacy of Zolgensma was demonstrated in key clinical trials, including the Phase 1 START trial and the Phase 3 STR1VE trial, which enrolled symptomatic infants with SMA type 1. The results were groundbreaking. Treated patients showed a significant improvement in survival compared to the natural course of the disease. In the STR1VE trial, 91% of patients were alive and free of permanent ventilation at 14 months old. Beyond survival, patients experienced rapid and lasting gains in motor function, reaching milestones like sitting without support, which is never seen in untreated SMA type 1. Long-term follow-up from the START trial indicates these effects are lasting, with patients maintaining motor milestones nearly 4 years after receiving the single-dose treatment. The systemic administration of a high dose of AAV vector poses significant safety challenges, mainly due to the host immune response. The most serious risk linked to Zolgensma is hepatotoxicity, including cases of acute severe liver injury and liver failure, which can be fatal. Therefore, patient management involves careful monitoring of liver function, aminotransferases, before and after infusion. All patients must receive a prophylactic course of systemic corticosteroids, such as oral prednisolone, starting 1 day before the infusion and continuing for at least 30 days to help prevent immune-mediated liver injury. Other notable adverse events reported include transient thrombocytopenia, low platelet counts, and increases in cardiac troponin I, necessitating monitoring of platelet levels and cardiac health. Elevidys is the first gene therapy approved for the treatment of Duchenne muscular dystrophy, a disease that posed significant challenges to the adeno-associated virus platform due to the enormous size of the causative gene. Duchenne muscular dystrophy is a severe, progressive, and inherited disorder that affects approximately 1 in 3,500 boys. It is caused by mutations in the gene responsible for producing dystrophin, a vital structural protein in muscle cells. Dystrophin functions as a shock absorber, protecting the muscle cell membrane during contraction. Without dystrophin, muscle fibers become susceptible to repeated injury, leading to chronic inflammation, muscle degeneration, and replacement of muscle tissue with fat and fibrous tissue. This ongoing damage results in a continuous loss of muscle function, typically causing patients to lose the ability to walk in their early teens. Subsequently, they often experience progressive heart and lung failure, which can lead to premature death. The full-length gene responsible for this disorder is among the largest in the human genome, with a coding sequence of approximately 11,000 base pairs. This size greatly exceeds the packaging capacity of adeno-associated virus vectors, which is around 4,700 base pairs. Elevidys overcomes the AAV packaging limit through clever bioengineering. It uses an AAVrh74 vector to deliver a transgene that encodes Elevidys microdystrophin, a shortened yet functional version of the dystrophin protein. This engineered microdystrophin is only 138 kDa, compared to the 427 kDa full-length protein, allowing it to fit within the AAV vector. It is designed to include the most vital functional domains of native dystrophin and has been shown to correctly localize to the sarcolemma, where it can help protect muscle cells from contraction-induced damage. The therapy is administered as a one-time intravenous infusion. The AAVrh74 vector was chosen for its strong affinity for skeletal, cardiac, and diaphragm muscles, and the muscle-specific MHCK7 promoter is used to drive targeted expression of the microdystrophin transgene in these tissues. The path to approval for Elevidys was notable for its complexity. In June 2023, the FDA granted accelerated approval for ambulatory pediatric patients aged 4 through 5 years. This decision was based on data showing that Elevidys increased the expression of microdystrophin in skeletal muscle, a surrogate endpoint deemed reasonably likely to predict clinical benefit. This pathway was pursued after the pivotal Phase 3 EMBARK trial failed to meet its primary functional endpoint, a statistically significant improvement in the North Star Ambulatory Assessment (NSA) total score at 52 weeks compared to placebo. However, the FDA noted compelling evidence of clinical benefit in secondary and exploratory endpoints, including statistically significant improvements in time to rise, TTR, from the floor and the 10-meter walk/run, 10 MWR, test. Based on the totality of the evidence from multiple studies, in June 2024, the FDA converted the accelerated approval to a traditional approval for ambulatory patients aged 4 and older and expanded the accelerated approval to include non-ambulatory patients. The safety profile of Elevidys highlights the difficulties of systemic AAV delivery. Common adverse reactions include vomiting, nausea, fever, and temporary thrombocytopenia. More serious risks require careful management and ongoing monitoring. These involve acute severe liver injury, which necessitates a preventive oral corticosteroid regimen for at least 3 months after infusion and weekly liver function tests. Cases of immune-mediated myositis, muscle inflammation, and myocarditis, heart muscle inflammation, have also been reported, requiring monitoring of cardiac troponin I levels. Importantly, Elevidys is not recommended for patients with a deletion in exon 8 and/or exon 9 of the DMD gene due to an increased risk of a severe immune response against the microdystrophin protein. Bugvez, approved by the Food and Drug Administration in April 2024, is the second gene therapy available for the treatment of hemophilia B. It provides a one-time treatment option that eliminates the need for lifelong prophylactic protein infusions. Hemophilia B is an inherited bleeding disorder caused by mutations in the gene responsible for producing a blood clotting protein called factor IX. This disorder is inherited in an X-linked recessive manner, meaning it primarily affects males. The deficiency of factor IX impairs the blood's ability to form clots effectively, leading to spontaneous or prolonged bleeding episodes. These episodes often occur in the joints and muscles, and recurrent bleeding into the joints can eventually result in joint damage known as arthropathy. The current standard treatment for individuals with moderate to severe hemophilia B involves regular intravenous infusions of factor IX, which can be either recombinant or derived from plasma. This prophylactic approach aims to prevent bleeding episodes and manage symptoms effectively. Bugvez is an in vivo gene therapy designed to enable patients to produce their own factor IX protein continuously. It uses an AAVrh74 VAR capsid vector delivered as a single intravenous infusion to transport a functional copy of the F9 gene to hepatocytes in the liver, which are the natural site of factor IX synthesis. A key innovation in Bugvez's design is the use of a high-activity variant of the F9 gene, known as the factor IX, or 338L or factor IX Padua variant. This naturally occurring variant encodes a factor IX protein with approximately 5 to 8-fold higher specific activity than the wild-type protein. This allows the therapy to achieve clinically meaningful levels of coagulation activity with lower levels of protein expression and, consequently, a potentially lower vector dose, which may improve the therapy's overall safety and efficacy profile. The approval of Bugvez was based on the pivotal Phase 3 Benegene 2 study, which assessed its efficacy and safety in adult males with moderately severe to severe hemophilia B. The study achieved its primary endpoint, showing that a single infusion of Bugvez was better than routine factor IX prophylaxis in lowering the annualized bleeding rate, ABR. During the efficacy evaluation period, patients treated with Bugvez had an average ABR of 2.5, compared to 4.5 during their initial prophylaxis period. Notably, 60% of patients had no bleeds after treatment, compared to only 29% in the prophylaxis group, and the median ABR was reduced to 0. These findings indicate that the therapy offers sustained, reliable bleed protection that can relieve patients from the burden of regular prophylactic infusions. A key step in patient selection is screening for pre-existing neutralizing antibodies to the AAVrh74 VAR capsid. Patients with significant antibody levels are ineligible for treatment. The main safety concern is the risk of an immune response against the transduced liver cells, which appears as an increase in liver transaminases, e.g., ALT/AST. This is the most common adverse reaction and is usually managed with a tapering course of corticosteroids. Patients need regular monitoring of liver function tests and factor IX activity levels after treatment. Additionally, long-term monitoring for hepatocellular carcinoma through abdominal ultrasound is advised for patients with pre-existing risk factors for liver cancer. The progression from Luxturna to Bugvez demonstrates a clear development in the field of in vivo genetic medicine. Luxturna served as an important proof of concept, showing that AAV-mediated gene replacement could be safe and effective in an accessible, immune-privileged organ like the eye. Zolgensma expanded this idea to a systemic level, addressing a severe neurological disease and proving that AAV vectors could cross the blood-brain barrier. However, this also revealed the challenge of systemic toxicity, especially hepatotoxicity. Elevidys tackled a different fundamental obstacle, the AAV packaging limit, with an innovative bioengineering solution using microdystrophin. Its complex regulatory process, including an initial accelerated approval based on a surrogate endpoint, also expanded the boundaries of regulatory science for this new class of medicines. Lastly, Bugvez further refines the technology by incorporating a high-activity, transgene, factor IX Padua, optimizing the therapeutic index to potentially achieve better clinical results at lower, safer vector doses. This step-by-step progression, from localized proof of concept to systemic delivery, overcoming packaging limitations, and enhancing transgene function, shows a field that is continuously learning from its successes and challenges to develop more advanced and effective therapies. While gene replacement therapies have been transformative for certain monogenic diseases, they mark the beginning of the story of genetic medicine. The next frontier is in vivo gene editing, a technology that aims not just to add a faulty gene, but to permanently fix it at its source within the patient's own genome. This method depends on programmable nucleases that can be directed to specific DNA sequences to create a double-strand break, DSB, prompting the cell's own DNA repair mechanisms to make the desired change. Three main types of engineered nucleases have been developed for gene editing: zinc finger nucleases, ZFNs, transcription activator-like effector nucleases, TALENs, and the clustered regularly interspaced short palindromic repeats, CRISPR/Cas system. Although all three induce a targeted DSB, they differ fundamentally in how they recognize their DNA target site. ZFNs were the first customizable platform for targeted genome editing. They are synthetic proteins formed by fusing two functional domains: a DNA binding domain made up of an array of zinc finger proteins and a DNA cleavage domain from the foci restriction endonuclease. Each zinc finger is engineered to recognize a specific 3-base pair DNA triplet. By linking multiple modules, a ZFN can be designed to bind to a longer, more precise DNA sequence, e.g., 9 to 18 base pairs. The foci nuclease works as a dimer, meaning that a double-strand break, DSB, occurs only when two separate ZFNs bind to opposite DNA strands in the correct orientation and proximity, allowing their foci domains to assemble and cleave the DNA. This dimerization requirement increases specificity. However, designing effective ZFNs is notoriously challenging and labor-intensive because the binding of one zinc finger module can unpredictably influence the specificity of adjacent modules, a phenomenon known as context-dependent effects. TALENs represent an advancement over the ZFN architecture, employing a more modular and predictable DNA binding domain. Like ZFNs, they are fusion proteins that utilize the foci nuclease for DNA cleavage. However, their DNA binding domain is made up of an array of transcription activator-like effector, tail, repeats, which were originally discovered in plant pathogenic bacteria. The main advantage of tails is their simple recognition code. Each repeat recognizes a single DNA nucleotide, and this recognition process is largely independent of neighboring repeats. This makes the design and assembly of custom TALENs much easier and more reliable than for ZFNs. Similar to ZFNs, TALENs function in pairs to enable the dimerization of the foci domains and induce a DSB. The discovery and adaptation of the CRISPR/Cas9 system, originally a bacterial immune system, revolutionized the field of genome editing due to its unmatched simplicity, efficiency, and versatility. Unlike ZFNs and TALENs, which depend on engineered proteins to recognize DNA, the CRISPR/Cas9 system employs an RNA-guided mechanism. It has two main components: the Cas9 nuclease, the molecular scissors, and a guide RNA, gRNA. The gRNA contains a sequence complementary to the target DNA site. It guides the Cas9 protein to that specific location in the genome, where the nuclease then makes a double-strand break, DSB. The strength of this system comes from its programmability. To target a new gene, simply synthesize a new gRNA with a matching sequence. This approach is far simpler, quicker, and cheaper than the complex protein engineering needed for ZFNs and TALENs, making gene editing technology accessible to nearly every molecular biology lab. The technological shift from protein-based DNA recognition in ZFNs and TALENs to the RNA-guided mechanism of CRISPR is the main reason for its widespread use and the surge in research in the field. While ZFNs and TALENs were powerful tools that introduced the idea of targeted editing, their dependence on custom protein design created a major barrier related to cost, time, and required expertise. CRISPR made the technology more accessible by transferring the complexity of specificity from a complicated protein structure to a simple, easily made RNA molecule. This key difference in how DNA is recognized explains why CRISPR is seen as a revolutionary, not just an evolutionary, breakthrough in biotechnology. The creation of a targeted DSB by a nuclease is only the first step in the gene editing process. The final outcome, whether a gene is disrupted or precisely corrected, is determined by the cell's own DNA repair machinery. Eukaryotic cells possess two major pathways for repairing DSBs: non-homologous end joining, NCJ, and homology-directed repair, HDR. Gene editing technologies are designed to hijack these natural processes to achieve a desired genetic modification. Non-homologous end joining, NCJ, is the primary and most active pathway for repairing double-strand breaks in the DNA of most mammalian cells. This process is rapid and efficient, functioning throughout the entire cell cycle by directly ligating or rejoining the two broken ends of the DNA molecule. However, the speed of this repair mechanism comes with a trade-off in accuracy. The machinery involved in non-homologous end joining often adds or removes a few nucleotides at the site of the break before the DNA ends are rejoined. This activity can lead to small, random insertions or deletions, which are collectively known as insertions or deletions. Researchers utilize the error-prone nature of non-homologous end joining to achieve gene knockouts. When a double-strand break is introduced within the coding region of a gene, it often results in insertions or deletions, known as indels. These indels can cause a frame shift mutation, which changes the triplet reading frame of the gene. This often leads to the formation of a premature stop codon, resulting in the production of a truncated, non-functional protein. Consequently, this process effectively disrupts the gene's function. Homology-directed repair is a DNA repair mechanism known for its high precision, although it occurs less frequently than other pathways. Unlike non-homologous end joining, which simply joins broken DNA ends, this repair process uses a homologous DNA sequence as a template to ensure accurate restoration of the original genetic sequence. In the context of gene editing, this mechanism can be harnessed by providing the cell with an external DNA donor template. This template contains the desired new genetic information and is flanked by regions of homology that match the DNA sequences on either side of the double-strand break. This approach allows for precise genetic modifications by leveraging the cell's natural repair pathways. Homology-directed repair is fundamental for making precise genetic edits. By designing a donor template carefully, this method allows for the correction of a single nucleotide mutation, restoring the sequence to a healthy state. Additionally, it enables the insertion of larger segments of DNA, such as an entire therapeutic gene, at a specific, predetermined location within the genome. This technique provides a powerful approach for targeted genetic modifications. A key challenge in therapeutic gene editing is the competition between two repair pathways inside cells. In most mammalian cells, NCJ is the default and dominant repair method, while HDR is much less efficient and mainly active during the S and G2 phases of the cell cycle when a sister chromatid is available as a natural template for repair. This makes knocking out a gene via NCJ relatively easy, but achieving high-efficiency gene knock-in through HDR is significantly harder. Current research focuses on developing strategies to favor HDR. These include temporarily inhibiting important proteins in the NCJ pathway, like DNA ligase 4, and methods to synchronize the target cell population cycle to increase the number of cells in the S/G2 phases, thereby boosting the chances of precise HDR-based edits. Building on the foundational principles of nuclease-based editing and DNA repair, the field of in vivo gene editing is now rapidly advancing into the clinic. While the first landmark approval for a CRISPR-based therapy was for an ex vivo product, a robust and growing pipeline of in vivo candidates is demonstrating the potential to treat a wide range of diseases by directly editing genes inside the body. This progression is marked by key technological trends, significant remaining challenges, and profound ethical considerations that will shape the future of medicine. The clinical pipeline for in vivo gene editing is expanding, with several companies advancing programs into human trials. These early-stage therapies are primarily focused on diseases where the target organ is the liver or the eye, as these tissues are more readily accessible for gene delivery. While the first CRISPR-based therapy to gain regulatory approval, Casgevy, exagamblogene autotemcel, for sickle cell disease and beta thalassemia, is an ex vivo treatment 68. The focus of many next-generation programs is on the more scalable in vivo approach. As of early 2025, approximately 250 clinical trials involving various gene editing technologies are being monitored, with over 150 actively recruiting or treating patients. Key therapeutic areas for in vivo gene editing include hereditary transthyretin amyloidosis. Intellia Therapeutics, in collaboration with Regeneron Pharmaceuticals, is developing a novel treatment called NTLA 2001. This therapy employs the CRISPR/Cas9 gene editing technology to disable the transthyretin gene in liver cells. The primary aim is to decrease the production and accumulation of the misfolded transthyretin protein that causes the disease. Notably, this was the first clinical trial to demonstrate successful systemic in vivo gene editing in humans using CRISPR technology. Hereditary angioedema is a condition that involves sudden episodes of swelling. Intellia's NTLA 2002 is an in vivo technology that uses CRISPR gene editing to target the kallikrein B1 gene in the liver. The goal of this therapy is to reduce the production of bradykinin, a substance that causes the swelling attacks characteristic of hereditary angioedema. In the realm of cardiovascular diseases, several companies are focusing on genes involved in lipid metabolism to develop treatments. Verve Therapeutics is leading the way with a technique called in vivo base editing, a form of CRISPR gene editing that makes precise single base changes without creating double-strand breaks. This approach aims to permanently inactivate genes such as proprotein convertase, subtilisin/cursin type 9, and angiopoietin-like protein 3 in order to lower low-density lipoprotein cholesterol levels. Similarly, CRISPR Therapeutics is developing therapies that target angiopoietin-like protein 3 and lipoprotein A, a variant of low-density lipoprotein involved in cardiovascular risk. Their investigational treatments include CTX310 and CTX320, respectively. Building on the success of delivering gene therapy using adeno-associated virus, Adidas Medicine is working on developing a new treatment called Adidas 101. This therapy utilizes the CRISPR gene editing technology and is delivered through the same type of virus to target genetic mutations. The focus of this treatment is to correct a mutation in the CEP290 gene. This mutation causes a severe form of inherited blindness known as Leber congenital amaurosis type 10. A key trend in vivo gene editing, especially for liver-targeted therapies, is shifting away from AAV vectors to non-viral delivery systems, mainly lipid nanoparticles, LNPs. These LNPs are tiny fat spheres that can encapsulate gene editing components, usually as messenger RNA, mRNA, for the Cas nuclease and the synthetic guide RNA, and deliver them to hepatocytes via intravenous infusion. This change is intentional. It is a vital adaptation of delivery technology to meet the safety and mechanistic needs of gene editing, which differ from those of gene replacement. Several reasons drive this shift. First, LNPs enable transient expression of the editing machinery. The delivered mRNA is translated into the Cas protein, which performs its editing function and is then naturally broken down by the cell within a few days. This hit-and-run approach is highly beneficial because it limits the duration the nuclease is active, reducing the risk of unintended off-target edits over time. In contrast, AAV vectors are designed for long-lasting episomal expression, which is ideal for gene replacement but can pose safety risks by allowing the nuclease to remain in the cell indefinitely. Second, LNPs tend to be less immunogenic than viral capsids and avoid the problem of pre-existing neutralizing antibodies that can hinder AAVs, potentially enabling redosing if needed. Lastly, LNPs do not have the same strict packaging limits as AAVs, offering more flexibility in the cargo they can carry. This strategic shift to LNPs for in vivo editing marks a major departure from the AAV-focused approach used in gene replacement, specifically aimed at improving the safety of permanent genomic modifications. Despite rapid progress, the widespread clinical use of in vivo gene editing faces significant challenges that need to be addressed to ensure safety and effectiveness. The primary safety concern is the possibility of the nuclease cutting at unintended locations in the genome, known as off-target effects. These unintended cuts can cause harmful mutations, gene disruptions, or even activate cancer-causing genes. Another concern is the occurrence of large, unintended on-target effects, such as sizable deletions or complex chromosomal rearrangements at the target site, which can also be harmful. A key focus of the field is to reduce these risks by developing higher precision Cas enzymes with less off-target activity and by improving guide RNA design for better specificity. The host immune system can also recognize both the delivery vehicle and the gene editing components as foreign, triggering an immune response that can compromise safety and effectiveness. Since the Cas9 nuclease is a bacterial protein, it can provoke antibody and T-cell responses in humans. This immune response can lead to the destruction of edited cells, decrease the therapy's effectiveness, and prevent repeated treatments. The production of clinical-grade gene editing components and delivery vehicles is a complex and highly specialized process. Ensuring consistency, purity, and potency on a commercial scale presents a significant manufacturing challenge. Additionally, regulatory agencies like the FDA continually update their frameworks to address the unique considerations of these therapies, including the permanence of genetic changes and the necessity for long-term safety and efficacy monitoring. The field is constantly evolving. The next generation of gene editing tools, such as base editors and prime editors, are already in clinical development. These technologies, derived from CRISPR, can make precise changes to the DNA sequence, such as correcting a single-letter mutation, without creating a DSB, which may provide a better safety profile by avoiding risks associated with DSB repair. Simultaneously, the integration of artificial intelligence, AI, and machine learning is set to speed up the development of safer and more effective therapies by enhancing guide RNA design, predicting off-target effects more accurately, and optimizing new delivery vectors. The ability to permanently change the human genome comes with a serious ethical responsibility. The discussion among the public and scientists about gene editing has centered on several main topics. It is important to tell the difference between two types of gene editing. The therapies that are in clinical trials now only use somatic gene editing, which changes the DNA of non-reproductive cells, like liver cells and blood stem cells, to treat disease in each patient. These changes are not inherited and are generally considered ethically acceptable for treating serious illnesses. This is quite different from germline gene editing, which involves changing the DNA of sperm, eggs, or embryos. Such changes would be passed down to future generations, effectively changing the human gene pool. Germline editing raises many complex ethical, social, and safety issues, and there is broad international agreement that it should not be used for reproductive purposes at this time. Because gene editing causes a permanent change to a person's DNA, the standard for safety is exceptionally high. The risk of unknown long-term effects requires thorough preclinical testing and long-term follow-up of clinical trial participants. A related issue is ensuring that patients can give truly informed consent. The complexity and novelty of the technology make it hard to explain all potential risks and benefits, especially when long-term effects are not yet fully understood. A major societal challenge is access. Gene therapies are now some of the most expensive medicines globally, with treatments costing millions of dollars per patient. There is a serious ethical concern that these potentially curative, one-time treatments will only be available to the wealthy or those in countries with advanced healthcare, which could worsen health disparities and create a new form of genetic inequality. Solving this problem will need innovative payment systems, public-private partnerships, and a global effort to make sure the benefits of this transformative technology are shared fairly. The field of in vivo genetic medicine has shifted from a distant goal to a clinical reality, fundamentally changing the outlook for many devastating inherited diseases. The first set of approved therapies, based on the AAV gene replacement platform, has provided clear proof of concept. Luxturna, Zolgensma, Elevidys, and Becvez have not only given patients life-changing benefits, but also established a practical therapeutic model, showing that a faulty gene can be effectively supplemented in vivo to restore normal function. This initial success story has been driven by repeated innovation, with each therapy overcoming unique biological and engineering challenges, from targeting an immune-privileged organ to achieving systemic delivery across the blood-brain barrier, to designing novel transgenes that fit within the vector's limits, and finally, to improving therapeutic outcomes with high-activity protein variants. Now, the field is on the brink of its next major evolution, shifting from gene replacement to in vivo gene editing. This transition promises a deeper level of intervention, moving beyond mere supplementation to the direct and permanent correction of the genetic code. The growing clinical pipeline, led by CRISPR-based technologies, is already exploring treatments for a wider range of conditions, including metabolic, cardiovascular, and eye diseases. This shift is driven by a crucial convergence of technologies. Improvements in nuclease engineering are producing editors with higher accuracy, lowering the risk of off-target effects. A strategic move toward non-viral delivery systems, especially LNPs, is improving the safety of in vivo editing by allowing transient expression of the editing tools. Additionally, the integration of artificial intelligence is set to speed up the design of more precise and effective therapies. Turning these powerful technologies into mainstream medicine will require more than just scientific breakthroughs. It will need a coordinated effort to overcome challenges related to safety, scalable manufacturing, and changing regulatory environments. Most importantly, it will demand a global commitment to addressing ethical issues like informed consent and fair access. If these challenges are met, the future of genetic medicine could go beyond treating rare monogenic disorders, offering one-time, curative solutions for a wide range of common and complex diseases, truly transforming the practice of medicine. This was an episode of the Nexus Clinical Podcast. This is for general informational purposes only and does not constitute the practice of medicine, nursing, or other professional healthcare services, including the giving of medical advice. No doctor-patient or pharmacist-patient relationship is formed. Using this information and the materials linked to this content is at the user's risk. This is not intended to substitute for professional medical advice, diagnosis, or treatment. Users should not disregard or delay seeking medical advice for any medical condition they have, and they should consult their healthcare professionals for any such conditions. Clinical experts created the references, content, and clinical insight. NotebookLM, a Google AI tool, created the content, which I extensively reviewed before release. Finally, the host states that he takes all conflicts of interest seriously. Currently, there are no conflicts to disclose. For all of his disclosures and the companies he invests in or advises, he directs users to reach out independently, where he keeps an up-to-date and active list of all disclosures.